Okay, so this was a play off the Supreme Court fiasco. Peter's talk titles. I literally just replaced beer with art. So, it's a bit of an outlier in that I don't have a lab. And I represent sort of the biological wing of Dan's lab. And Dan, one of the amazing things about Dan is the diversity of work he did. So I think at the same time he was, wherever we were publishing biology journals, he was publishing physical reviews letters, and it's just quite remarkable. And biology is tough because it's complicated and it's sloppy, and 95% of papers that are published in biomedical science are just fundamentally flawed, they're wrong, and not many people have the wherewithal and I think the guts to call people out on that. And so, two of the many things I really appreciate about Dan was just, I think he once summed it up as it's not about being right it's about getting to what's right, and then also, I know he's got a lot of flack for it, and a lot of people are scared of him for it, but just his honesty, we need more of that. So, anyway, when I joined the lab, I knew I wanted to work, when I came to Stanford, I wanted to work with Pat and Dan. I just didn't know how to make that happen. And they have embarked on some work to basically study gene expression programs. So Pat was, figuring out how gene expression programs work with the DNA microarray, how transcriptional regulation works, and I think Dan was going to RNA conferences and seeing people just do northern plots. They figured, okay, well, let's marry this beautiful relationship here and team up. So at the time, most of the regulation of gene expression was thought to be transcriptional, but there are lots of reasons they thought that was wrong. And I just want to highlight, just to mention, some of the early work that was done by Yule, systematically measuring RNA decay, Yoav with translation, and Andre with RNA binding proteins. And others did this sort of thing. They just didn't do it as well. And so I don't think they reached the level of understanding and were able to push the field that Dan and Pat were able to. Andre's work was really influential on them. So, nice picture, right? So anyway, I was having a blast with Dan and Pat, but then push came to shove, and I had some bills to pay. So I went and found a job. And so I moved family down to San Diego. So I was recruited to a company whose goal was to develop oligonucleotide therapeutics that target microRNAs. So these are small regulatory RNAs that are encoded in our genomes that play important roles in development and physiology. And the idea was built on basically taking an oligonucleotide that matches the microRNA, using a diversity of chemical modifications that you put on the oligo, and the fact that a single-stranded oligo, if you just inject it subcutaneously, will get gobbled up by plasma and delivered functionally to the liver and kidney and some of the other tissues in the body. And so that was the basis for one of their parent companies, Ionis, who used to devices. And these chemical modifications that confer drug-like properties, but they had sort of emergent properties to them. So it's hard, there are no rules as to how to make a good oligo that's going to have favorable properties. And so they wanted me to figure out how these things work and then use that information to improve upon them. So so turns out there are a number of steps in the microRNA maturation or that they could in principle work at. So they could either work during their biogenesis or also in the context of its mature effector complex, which is with an argonaut. And there is speculation that they stimulate the decay of the micro or but to make a long story short, it's pretty simple. They interact with the micro in the context of risk, and they start to block the risk. And so I use that information to basically, I reason that, okay, well, if it's acting like a mature target, it's acting in the context of argonaut, we can use the principles that have been worked out for microRNA recognition for designing the oligos. And that helped. And then move things forward a bit. And so we applied those designs to our lead program, which is for the microRNA miR-122, which is only expressed in hepatocytes and actually is a required host factor for hepatitis C virus. So basically, workloads, a lot of done by Peter Sarnow here, acts like a cat for the virus. So you get rid of miR-122, the virus can't replicate, and you can clear it. And at the time, well, the hepatitis C therapeutic landscape has changed tremendously over the last few years, but at the time there weren't good therapy options. So we applied those principles. It got us some good oligos, but they weren't good enough. They weren't drug-like. But the thing, they didn't have a high enough therapeutic index to take into humans for clinical trials. And the thing that changed that all, and the thing that's been a renaissance for oligonucleotide therapeutics in general, is the notion of using targeted delivery. So not just injecting and having them passively taken up by tissues, but putting on a molecule that will target them to a specific cell or tissue type. And the one that's really worked has been GalNAc, which is recognized by the acylglycoprotein receptor, which is only expressed on hepatocytes. It'll internalize its cargo and release it in the cytoplasm. And that whole mechanism was worked out, I think, in the late 70s, and people had tried to use it for drug delivery for quite a while and pushed it. And then finally, a gentleman who had been working on this first at ISIS and then out on Island for like 20 years got it to work on Island. We got that information, we synthesized it, and just like that, it shifted our potency of the compounds 20x in vivo. It shifted therapeutic index 100x. It was a real watershed for us, and it made a molecule that was good enough to go into clinical trials. And it's also enabled, I think, there's a whole generation now of oligonucleotide therapeutics that use this GalNAc that are going to finally make it over the approval line for the FDA. And so the drug went into clinical trials. It cured a number of patients with a single dose, which is pretty cool. After I left the company, went to phase 2, and I think it petered out. It was too little, too late. I left Regulus. Why did I leave Regulus? So things were going all right. But one thing I wanted to show you was this funny result I found, was that we screened compounds. So as I said, you just sort of have to, for the most part, just empirically screen compounds. There aren't much for design rules. So we screened compounds with this activity assay in cell culture. And I noticed, and I developed some assays to screen compounds for cytotoxic effects. And I noticed for this one microRNA that all the compounds that were the most potent in the activity assay were also the most cytotoxic in a different cell line. That's weird. So I asked if it extended to another microRNA, and it did. So I reasoned, well, there must be differences in transfection efficiency. There must be differences in them getting into the cell. And in fact, it is. transfection efficiency. And so most of the variability in our whole screening process was explained by differences in the oligos just getting taken up by lipofectamine. And it explained why, in part, why there was like no correlation or predictive value for cell culture systems. And so they didn't like this.
Can we just sort of like get it straight whether when you went to the CEO of the company and explained it, were you sort of like laughing like this, or were you like having more serious demeanor when you brought it up?
Well, I think it's as clean a result as you can get. It was just no one got it. It was just like, no. It was like for months, and I kept bringing it up, and I'm like, no, we're doing it wrong. We can fix this. Find another way to get them into the cell and find better assays. And finally, we're sick of this. You need to leave. I was like, well, you know, it's not just us. It's like all the other companies that are doing this. It's probably the same damn thing. They're just wasting millions and millions of dollars on this. But anyway, so they showed me the door. I'm hoping statutes of limitations have expired on that, so I'm okay showing you. So I went to a better place where I'm at now. It's called Hogan, and this is pretty cool technology. I'm just going to tell you a little bit about the technology in my last couple of minutes. And it involves tumor-selective replicating retroviruses. So we have simple gamma retroviruses that is our platform. And these basically will selective, and they're replicating. So we can inject these into a tumor or even IV. They will pretty much only infect tumor cells and only spread within the context of a tumor for a variety of reasons. So you can basically use this as a tumor selective gene delivery system. And so the lead, product or vector is this sort of old classic case of the pro-drug activator. So we have our replication system here, and then we have cytosine deaminase for yeast. So you inject this into the patient, spreads in the tumor, then the patient eats a pill of 5-fluorocytosine, which is an antifungal, and it gets converted by the cytosine deaminase into the cytotoxic molecule 5-FU locally in the tumor, which leads to both direct cell death killing and an immunological response. And so, as you probably there's been a renaissance in cancer therapeutics and our approach to attacking cancer through finally the ability, it's long been suspected that the immune system should be able to recognize tumors as being foreign because they're mutated, they're producing neoantigens, they're producing foreign molecules, but the tumor is very good at having a suppressive immune system and suppressing that effect. Jim Allison, got the Nobel Prize for his very nice work on the CTLA-4 on that. And so our drug also has an immune activator effect, quite distinct from the checkpoint inhibitors. And this is just showing that in mice, an immune deficient mouse, if you give the virus and then they eat the 5-FC, the tumor goes down and they'll come back. If it's immune competent, it'll clear it. And basically, this is just showing that it has a memory of it, and so the tumor can't get back. And if you take those T cells back, so the virus, it turns out that some of the key suppressor cells in tumors, the myeloid suppressor cells, are exquisitely sensitive to 5-FU, and so the 5-FU killing plus I think leads to a release of antigens. Plus probably also viral proteins probably being recognized as well. And also you're getting rid of those key suppressor cells, which then allows the T cells to come in and do their job. And that's sort of been a big block in the field, is how to work upstream of the checkpoint inhibitors. And so we're right now in a registrational phase 3 trial in recurrent glioblastoma. So glioblastoma is basically the worst of the worst. There's no cure. When you first get it, you have on average about 2 years to live. It inevitably comes back. When it comes back, you have on average 9 months to live. There are no molecular therapeutics approved for it. It's basically just chemo, radiation, and surgery still, and it's, lots of promising drugs just haven't made it, and there's a desperate need to sort of move the needle here. So, for a variety of reasons, we went after it, and this is just showing results with the phase 1 study, and so here's sort of historically how well patients do, and this is showing that on average, or the median, they're doing 50% better. It looks better, and then we have a swath of about a fourth of the patients show a nice response of the tumor regressing, and are also just still alive now. So at least 4 or 5 years out. So it's promising. Most of my work involves just sort of figuring out what distinguishes the responders from non-responders. Are there common underlying molecular features that we could use that tell us about mechanism of action of the drug, or can be used to segregate patient populations, or go into what other indications to go after. And this is, I'm over time, this is just showing the tumor will start growing back, and then it'll shrink again. So there's an immunological response that happens months after the treatment starts. And we have an ongoing phase 3 trial that's fully enrolled, and I find myself reverting to my Catholic youth. So I'm talking about the trial, and we'll see. Hopefully it moves the bar. So, a couple pictures here. One of the great things about my interactions with Dan was that not only did I get to work in the lab with him, but I also got to coach soccer with him there. That's our first team of Jaime, as I recall, as coach. There's Sam right next to Dan. We also used to trick-or-treat in Dan's neighborhood, so with very fond memories of that. And Dan had a very profound insight in soccer that I still, I think, predicted the modern game, which was we were down, I think, 1-0 at halftime, and he told the kids, what happens if they score another goal? I'm like, well, we're down 2. He's like, yeah, we're still losing. What happens if we score? We're tied. He's like, don't worry about them scoring, score. Which is like, it's like the pull the goalie argument. I think Pep Guardiola, I think acknowledged you recently.
Okay, time for 1 or 2 questions.
So for the regulates, because I keep wanting you to relive that experience. So what happened to the clinical trials? And was it just that the other drugs came in at the same time that were?
Right. So Gilead, it's a couple. So finally, it was figured out how to culture hepatitis C in culture, so you could actually dissect what the proteins are doing and develop small molecule drugs. So that happened, and it led to approval of Harvoni that cured patients. But Gilead's business model is to wait. At least in the U.S., they can get away with charging like $1 gazillion for this, and so we're never going to eradicate hepatitis C that way. It's also criminal. But so there was an interest to try, that's usually like a 12-week cycle. So we figured our one shot, because we took too long getting there, was, okay, you take a single dose of this and then a 4-week with that, and you should be cured. And that worked. I think, again, I wasn't there, but I think they didn't do. I think they messed up on the dosage. It looked to me from afar that they're going higher than they needed to, and they also ran into some. So they had some safety issues. I don't know if it was on target or off target, and like, you know, after I left, the company's been straight downhill. So I look. Well, it has been, but it probably has nothing to do with it. Yeah, it was too little, too late. You know, it was. I think if it had been perfectly clean, maybe it had a chance. You know, hepatitis C, we talk about eradicating it, but so a large fraction of the population of hepatitis C are either in jail or homeless. So I'm not going to make a joke about them being poor and difficult, but you know, you need something that's cheaper and that I think is going to be easier than a 12-week cycle. Or Gilead to change their business model.
To go to the next speaker. Oh, you want? Is there a quick question?
I was just curious what in the non-responders case, and it seems like that happens several times. So I know the poliovirus trial from Duke and see the same type of studies. So what is your best guess at this point? Why only 20-30% show this prolonged, although remarkable response?
Right. So yeah.
Any ideas?
So I mean, the hard thing about this and trying to find predictive biomarkers for a therapy like this is there's so many different steps where it could fail. So I mean, one requisite is probably the immune composition of the tumors. So that looks to differ significantly between the responders and non-responders. So that's an important one.
But irrespective of the virus that you use?
Well, I don't know
Do they have
because
cross compare them?
well, one. So So that's another. You know, clinical trials you just don't share the data, right? It's frustrating, and so I don't. We've tried. Hogan does a pretty good job of publishing and trying to, you know, get out the molecular information so others can look at. They haven't with Duke, so we'll see. It's
It'll be
but it's a
interesting to see
strikingly
if
similar
Yeah.
distribution to ours. So
Yeah, that's what
it's
struck me.
similar type of therapeutic. So you think there are probably common features among those survivors, long-term survivors, or not. I mean, it's hard with phase 1 too. You know, I mean, better surgeons are probably influencing things. I mean, probably the most important predictor, arguably, is like who your wife and family is. Like, real. I mean, you know, there are a lot of variables. I don't know.
Okay, thanks. Then again,